Well, thank you for having me. It looks like everybody can see my screen okay. Cool. So my name is Jim. I'll be presenting our work today on sylph, which is a new metagenomic profiler, and now it's published in Nature Biotechnology. So this presentation will be about metagenomic profiling, so not 16S sequencing. The workflow looks like this. You have some microbiome, we have some metagenomic shotgun reads, and then profiling is using a database of genomes to infer the presence and abundance of taxa, right? So you might be thinking, why build a new metagenomic profiler? You probably have heard of these things like Kraken and Metaflam before, and they're very good. But the reason is that it's actually a really hard technical problem to classify reads when you do metagenomic profiling. You can imagine that indexing hundreds of thousands of genomes and mapping all these reads is very expensive. And I'm sure you've encountered this before. And also, if you've used some of these tools, you've noticed probably that there's a lot of false positives in these tools. So there's still work to be done here. In this work called sylph, we kind of do the inverse problem of classifying reads. What we do is we classify genomes against reads instead. I'll explain how you do this, but what we do is we check if each genome in our database is contained within our reads. And how we do this is we start off with something called k-mer sketching. You might have heard of something like sour mash before, which is very similar. And what we do is we look at the k-mers within sequences, which are these k-length strings. And then sketching is just subsampling the k-mers to increase the speed. So you just take a subset of the k-mers. And once you have the k-mers, what sylph does is it looks at k-mer containment. So if you have a database genome here, you can sketch the k-mers, obtain a subset of the k-mers. You can do the same for your reads, your sequences, all of your reads at once. You can just check if the k-mers in your genome are within of all of your reads, right? And this is some sort of notion of containment. And what we do specifically is use this notion of containment, average nucleotide density. You might have heard of ANI before in the context of comparing two genomes, but what I want to do is I want to compare a genome to a metagenome. And in this little graphic here, I have a reference genome and I have a similar genome in my metagenome, as well as some unrelated species. And the k-mers are slightly different because the genomes are different. So what you can do is you can apply this formula, which tells you that the ANI of the genome to the metagenome, this containment ANI, is a function of the fraction of contained k-mers. I won't go into this formula in detail, but you can go from k-mers to ANI is the main takeaway here, which is really nice. And why it's really nice is because I'm sure some of you have heard of this before, which is that we say that 95% nucleotide density is a sort of species level cutoff. So what sylph can do is you can check the containment ANI towards your metagenome and say if it's more than 95% similar, then it's present at the species level. And this is kind of a graphic showing how it works. So people have done this before. This is not new, using these k-mers to do containment. But what is new is tackling this low coverage sequencing problem, which I'll explain now. So the picture I showed you before wasn't completely accurate, right? So if I have a metagenome, I don't have all of the genomes. I have sequencing of the genomes inside my microbiome. And what that means is that some of these k-mers will not be sequenced because if the genome is low abundance, it will have low coverage. So I may not be able to sequence it. So that formula I presented before doesn't work because this B over here, this metagenome is undersequenced. So what sylph does is we use some statistical methods to correct for this. I don't have time to go into all the methodological detail. You can check a paper, which I'll link at the end. But the general idea is that if you know something about DNA sequencing theory, it's connected to Poisson statistics for how we calculate coverages. And this can be used to correct, unbias that ANI that I talked about before, which is this formula down here. So I'll jump into the results now. You may have heard of this CAMI2 Marine Challenge before. CAMI2 is this general suite of standardized metagenomic benchmarks. And over here, sylph is highlighted. It works well in this marine species level profiling test. But what I think might be interesting to all of you is how fast sylph is. It's really fast and efficient. For multi-sample profiling, if you're profiling a lot of samples at once, sylph on our data set only took about 2 seconds per sample. And it took 30 times less memory than Kraken. So I encourage you to try it out. Oxford Nanopore independently benchmarked sylph, so it works for long reads too, and they found it was very effective on their microbiome data set. So I'll quickly recap now. Sylph is a new metagenomic profiler. What it does is it classifies genomes against read using k-mers and some statistics. And it's really, really fast and efficient. But there are drawbacks. You cannot classify reads using sylph. Some tasks, if you only have one read, for example, you need to run something like Kraken. Sylph really requires a little bit of higher coverage than something like that. All right, so our paper is out now on Nature Biotech. You can go look at it. This is work done by me. I'm a postdoc at Harvard and Dana-Farber now with Professor Hung Lee. I did my PhD at University of Toronto with William Yu, who's now at CMU. And you can go on to GitHub and try out sylph. There's a few commands. You can start profiling against 100,000 genomes pretty quickly. Thank you.